I won't repeat anything I already told you last time. So you remember the importance about cardiovascular disorders. Suppose you take an adult man or an adult mouse. You can dissolve the heart in solutions and put in culture cardiomyocytes. There are efficient procedures to do so. You recover these very big cells. So the nucleus is about the same size as the nucleus of any other cell in the body. However, look at how big the cytoplasm. And the cytoplasm is often binucleated. These striations are the contractile apparatus. These are beautiful brick-shaped cells. And you can have these cells in culture, do electrophysiology, do a lot of molecular biology, in situ studies, whatever you want. For 40 years, people have tried to have these cells replicate, always failed. They don't replicate at all in culture. If you take bromodesoxyuridine, you heard already in the previous talk that you can use this compound to label a replicating cell. You inject bromodesoxyuridine into an animal. You look how many cells replicate in the heart. The answer is 0. You can see some replicating cells, but these cells are always outside cardiomyocyte, which are stained here with an antibody against alpha-actinin in green, and are cells of the endothelium or cells that travel through the blood vessels. Cardiomyocytes replicating 0. So the question is really the heart a completely post-mitotic organ, as you could guess from this snapshot of what happens at one time? But if you look at over time, is it really a post-mitotic organ? And there's been a fantastic study a few years ago of a group from the Karolinska Institute in Sweden who took advantage of the fact that you can measure now by sophisticated physics machineries called accelerator mass spectrometers the exact amount of C14 that you can find in any kind of samples. And you know the C14 is an isotope of carbon, which is very, very low. And except that during these 60s, there has been an enormous increase of the amount of C14 in the atmosphere due to the fact that the big superpowers in the United States, Russia, France also, started to explode bombs in the atmosphere. And these bombs produce a lot of isotopes. And at a certain point, this nuclear experiment was banned. There was a famous agreement between USA and USSR at that time that banned these kind of experiments. And so from that time on, the amount of C14 started to decay. Then now we are 6 or 7 years from the levels going back to normal levels. But since all the biochemistry of living beings is based on chemistry, then from the amount of C14 which is incorporated in macromolecules, you can judge whether that macromolecule was formed in the 60s or was formed, for example, the week before or in our era. So, for example, if you take the DNA and you measure the amount of C14 in DNA from gut cells, you would expect that these gut cells have been formed in the last week or 7 days. So the question was in this beautiful paper that was published in Cell, as I was saying a few years ago, whether the amount of C14 in the cortex was the amount of C14 that was present in the atmosphere when the person was born, or the amount that was present when the analysis was done, for example, in a 60-year-old individual. And the answer was the amount was exactly that of the age when he was born. And this was a clear-cut demonstration that basically our neurons are post-mitotic. The same group then repeated this experiment in 2009 in cardiomyocyte, and the answer was that the number of cells that are produced in the postnatal life, so in the adult life, in a man of 75 years, are less than 50% of the cells in the heart, which means that more than 50% of the cells in the heart are exactly the same cells with which we were born, which is remarkable because these cells work physically, so they keep contracting about 3 billion times over a lifetime and still are exactly the same cell with which we are born. This is basically a uniform view of most of the laboratories in the field. There are, I have to say, some exceptions. For example, the group of this scientist, which is now in Harvard, Pierre Anversa, believes that there is a more remarkable turnover of cells in the heart because there is some apoptosis, some cell renewal, so there is still some debate. But I would say that the broader view in the field is that the heart is really a postmitotic organ. Everybody, however, agrees that even if there might be some more renewal in physiological condition, everybody agrees that after myocardial infarction, repair is never by regeneration of cardiomyocyte, but it's always through production of a scar. So basically, the heart heals itself not by producing new contractile tissue, but generating fibroblast and collagen, so producing a scar. And the presence of the scar obviously reduces on one side, on one hand, the amount of contractile tissue. On the other hand, also imposes a mechanical stress to the rest of the myocardium that contracts, and this eventually is the major cause of heart failure, of which we spoke last time. 15 million people dying every year, 80% in developing countries. Drugs that have been produced, the last category of drugs, more than 20 years ago. So this is the reason why we have to find new ways of regenerating the myocardium. And these new ways cannot be a molecule, a chemical molecule, but most likely should be a biological molecule, so a gene, a protein, a factor, or a cell. Last time we spoke about how to find possible proteins that perform this job. Now the topic of this talk will be on how to find cells. An interesting question is, but if the heart has to be formed, so in the embryo there must be replicating cells. So in the adult there are no replicating cardiomyocytes. There must be some point when replication of a cardiomyocyte stops. When this point is. And if you do the same BRDU experiment, instead than in adult mice, in neonatal mice, so if you inject BRDU immediately after birth, surprisingly, you will see that almost 40% of the cardiomyocytes are still replicating. So 40% means that the whole heart keeps replicating in the embryo up to birth, and in 1-2 days in mice, 1 week in rats, a couple of more weeks in humans, then cardiomyocytes withdraw from the cell cycle, will never replicate again. So there is a short time window immediately after birth when cardiomyocytes are still capable of replicating, then they go out from the cell cycle. The reason why they do so is completely unknown. There are 2 major changes occurring at birth. One is the oxygen tension. So the heart is a venous organ in the womb during pregnancy because it's very far from the lungs of the mother. And so you receive deoxygenated blood, while it becomes flooded with oxygen from the lungs immediately when the neonates start breathing. And this is a major change that could impact on the proliferation capacity. And the second is the pressure to which it's subjected, because all the circulation in the mother is provided by the heart of the mother. The circulation of the newborn also has to be supplied by the pump function of the newborn. So probably one of the stimuli dictate the fact that cardiomyocytes stop dividing, and then from that time on, all the growth in size of the heart from the neonate to an adult occurs through increase in the expansion of the cytoplasm, so a mechanism called hypertrophy of the cardiomyocytes. You can reproduce at a certain extent this capacity of neonatal cardiomyocyte to replicate and then get out on the cell cycle also in culture. So basically you can take cardiomyocyte day 1 after birth, you will find cells that are round. They're already cardiomyocyte because they express marker cardiomyocyte like alpha-actin in green here. And then if you wait 3 days or 7 days, these cells becomes much bigger, so they hypertrophy, they organize the contractile apparatus, and become striated, and they get out of the cell cycle. So the number of cells in the ESA very significantly reduced. Please pay attention, because this will become relevant later in my talk, that if you take these day 1 cells, these are not stem cells, progenitor cells, but they are real cardiomyocytes, because they have differentiation markers, so they have a contractile apparatus, which is already there. They are big cells, but still these cells can replicate. So you see they are positive for cyclin A, which is a marker the S phase, they incorporate BRDU, or you can even see mitotic figures. So these are real cardiomyocytes that replicate. They're not dedifferentiated cells. People have tried again for years to have these cells proliferating indefinitely. They have expressed the T antigen, c-Myc, all these growth factors, the cyclin, the CDKs. Some of these treatments can indeed stimulate the cells entering S phase, so they start proliferate, they start duplicating their DNA. They can have even karyokinases, but when the cells have to undergo mitosis, there is a mitotic catastrophe. The cells die by apoptosis. No way of prolonging proliferation of these cells. Another experiment is very interesting to prove that really the heart has a very limited replication capacity. Was published this year in January in Nature. This is a group, again, from Harvard, actually, with a sophisticated method of mapping the replication capacity of cardiomyocytes in an adult mouse. Basically, they proved that there is a very, very limited capacity of proliferation, very compatible with this less than 50% of renewal over a lifetime. The other thing that this study did, which is very interesting, is that this very limited capacity of proliferation is not because there are stem cells that replenish the heart and have a cycle by which they differentiate into cardiomyocyte, but it is sustained by the cardiomyocyte themselves. So there is a very, very limited capacity of cardiomyocyte of replicating in the adult life, so limited that it becomes completely irrelevant if there is a big portion of the myocardium which is lost because of an infarction. So how can we overcome therapeutically this problem? That is, how can we stimulate cardiomyocyte proliferation in mammalian cells? And obviously, the first question that comes to everybody's mind is, let's use stem cells. Now, we are living in an era of stem cells. You can't read any newspaper without the concept of stem cell is somehow put forward. And so people have started, obviously, to think of stem cells for regeneration of the myocardium. Actually, the first cell type that people started thinking was not really stem cells, but stem cells from the skeletal muscle, so myoblasts, so satellite cells and myoblasts from the skeletal muscle. This is a very weird thinking, so, but gives you the medical need. There is such a medical need for heart regeneration that at the beginning people said, well, the heart contracts, the muscle contracts, they are both muscle tissues. Well, what's the difference? And so there have been trials performed by taking skeletal myoblasts and injecting these myoblasts into the heart. There was a group, especially in Paris. Menachet is the person who led the studies, which they took muscle biopsies from patients, expanded the myoblasts in culture for 2-3 weeks, and then injected these cells into the heart. There were companies that were established. They were selling myoblasts from other people to be injected in the heart. The result was a disaster. These myoblasts got into the heart, but they never integrated with the cardiomyocyte cells of the heart, because cardiomyocytes form a functional syncytium by which there is a single cells are individual, but they are coupled through a specialized type of gap junctions where there is a protein called connexin 43 that permits a very rapid passage of the electric signal. This is the reason why there is a pacemaker cell who starts contracting and gives an electric signal that synchronously goes through the myocardium, and this is the reason why all the cells contract simultaneously, and this gives the pump function. If you inject any kind of other cell types inside the heart, then these cells don't connect through these connexin 43 gap junctions, and so they create an electric barrier, so there can be other cell types that start giving a pacemaker signal, and so basically there are a lot of arrhythmia. People injected started to die because of these treatments. Now all they were taken back to the hospital and implanted with artificial defibrillators that can become active in case of ventricular tachycardia, which is ventricular arrhythmias, which are lethal. So forget about this kind of treatment. Then, at a certain point, about 10 years ago, there have been a flood in the scientific literature of papers reporting that the bone marrow might contain some cells that in special condition could become any kind of other cell types in the body. And the fact that there are cell types with stem cell property in the body is not new, because we knew for several years, for example, that the bone marrow contains hematopoietic stem cells that are used for bone marrow transplantations in people with leukemia, and these cells are really stem cells because they can self-renew. And at the same time, their progeny can become a red blood cells, a white blood cells, a platelets. So all cells that circulate in the body comes from a single cell. There was a fantastic paper 15 years ago in Cell in which you could take a single cell from the bone marrow and rescue completely a mouse from death by irradiation. So proving that a single cell can reproduce completely the bone marrow and all the blood in a living organism. And then it is known that the skin contains stem cells that reproduce the epidermis, and it is known that in the skeletal muscle there are the satellite cells which can reproduce the muscle. So these are all cells that are specialized to each one to give rise to a defined progeny for that specific tissue. But at a certain point, there were a number of reports published in the big literature that the bone marrow might contain cells that not only can reproduce are stem cells from the hematopoietic system, but also stem cells for other organs. For example, these are 3 papers in which bone marrow cells were reported to regenerate the infarcting myocardium. This is the most famous of this experiment. It's again from the Piero Anversa laboratory, and basically this is a Nature paper 2001, in which the author reported that you could reform almost 70% of the whole myocardium simply by injecting stem cell derived from the bone marrow inside the myocardium. 70% of the myocardium, and regenerating the myocardium 70% means regenerating cardiomyocyte, but also regenerating blood vessels, regenerating the architecture, regenerating the stroma, the fibroblast. And these are just 3 papers from a myriad of papers that were published around that time, in which basically cells from virtually all kind of tissues could become cells of virtually all the other kind tissues. So muscle that becomes blood, muscle liver that becomes bone marrow, pancreas that becomes liver, bone marrow that becomes heart, and then bone marrow become neurons, but also neurons that become blood, neurons that become bone marrow. So basically, the idea was that there are some cell types in the circulation that are just traveling in the blood as a highway, and then according to the exit where they take, they can become any kind of cells to regenerate all organs. That was a very, very exciting period, and clinical people took this information by granted. So a lot of trials started for several organs. The cardiologists are one of the most aggressive in this kind of intervention. First, because there is really a medical need to regenerate the heart, and second, because reaching the heart is very simple. So clinical interventional cardiologists have several ways to reach the heart from the outside by thoracotomy or during heart surgery for bypass surgery, or they can reach the coronary artery by catheterization. They can reach the coronary sinus, which is the venous part where all the venous the venous blood from the heart goes. You can reach the inside of the myocardium through catheters, so it's very easy to reach the heart and inject something in the heart. So there's been a very aggressive recruitment of patients who have bone marrow stem cells, injected stem cells in, now we see in a moment in brackets, injected in the heart, hundreds of patients, thousands of patients. At the end, there was even advertisement in the internet for people who wanted to be enrolled in this kind of trials. After a few years, the results were published and were very, very, very disappointing. This is the first, this is a review from the New England After 2-3 years, this trial was started. Here we are at the end. We are about 6 or 7 years ago. And you see these are the first 5 clinical trials that were controlled with placebo. And you see that the results were really disappointing. For example, here, no difference between the two groups, no significant difference, no significant difference. Those who give a difference is just a small difference in performance of the heart seen by echocardiography, which is not at all it is a very subjective way of measuring cardiac functions, and with a few percentage points, which probably in clinical terms means nothing or little or close to nothing. At the same time, people try to reproduce these first experiments of trans-differentiation of stem cells from the bone marrow, or plasticity of bone marrow cells, as you want to call them, and these results failed. They failed because somebody showed that these initial exciting results were not due to real proliferation of a stem cell and differentiation, but because some cell that you injected physically fused to cell from the organ. So bone marrow cell that fuses for an already existing cardiomyocyte. So you have the impression to have a cell that comes from the bone marrow and becomes a cardiomyocyte, but instead this is due to the fact that you have a cell that has 2 times the amount of chromosome, a cell that does not divide, and so cannot be responsible for regeneration. So this is called fusion. And at the same time, other groups try to reproduce the first experiment in mice and completely failed. So you see these are 2 back-to-back papers in Nature again. And one says that hematopoietic stem cells do not transdifferentiate. The other says that hematopoietic stem cells are stem cells, but when they are injected in the heart, they continue to do what they know, so to produce blood, but they don't become cardiomyocytes. And now I have to say that this whole concept of plasticity is something that in very, very few laboratories still continue to believe that it really exists. Very few laboratories continue to believe that the bone marrow might contain cells that can become in any kind of cell types. All the experiments that take some types of bone marrow-derived cells, like mesenchymal stem cells, and see that these cells in the laboratory become cartilage, bone, skeletal muscle, cardiomyocyte, any kind of mesenchymal types of cells are true in the laboratory, but are never being reproduced when the cells are injected in vivo. So there is much skepticism about this broad presence of the so-called adult stem cells. The trials in the heart, however, no, let me say one thing more. Remember, please, that the only clinical applications of stem cell that holds true now are application in the bone marrow for bone marrow transplantation. Here, really, there is a stem cell that can become all cells circulating in the blood, and a couple of applications for epithelial cells, one in the skin. So if you have people with big burns, a lot of the proportion of the skin is burned, then you can save these people by taking a small portion of their normal heart and growing a thin layer of keratinocytes, and then apply this layer to the wounded area. So there must be a stem cell from that forms keratinocytes in culture, and this is really a stem cell, but it is so restricted that it can produce only the epidermis and not the dermis, so not even the fibroblast. And a variation of this is if you take cells from the limbus of the cornea in people who have a corneal opacity that makes them blind, because the cornea is the transparent layer in front of the eye. These are people who are blind. The only way to treat these people is by corneal transplantation from cadavers. Alternatively, you can take a piece of the limbus and grow these epithelial cells to form a transparent layer in culture, and then you can implant this layer in front of the eye, and these people regain their capacity. Also, in this case, it is a stem cell which is very specialized to do exactly that cell type, so not a broad, generalized stem cell. If you look at all the meta-analysis of all the trials that have been conducted, this line here is no effect. This is worse, this is better. You see that most of the trials are slightly better, so slightly that this cannot be due to regeneration, but probably is due to the fact that when you inject bone marrow cells, these cells start to secrete some factors, like angiogenic factors or microvesicles or growth factor that somehow sustain cardiac function. So there is a very, very limited benefit from injecting cells in the heart, no proof at all that there is regeneration. Nobody in the big clubs now believes that this is regeneration. There are some laboratories that still believe that some cell types with a special phenotype might be involved in regeneration, but a definite proof is really missing, and most disagree with these kind of findings. Can we use some other kind of stem cells for regeneration? There are real stem cells. The real stem cell is the cell that comes from the embryo. So after the zygote is formed by fertilization of an oocyte with the spermatozoon, then this zygote starts dividing to cell, 4 cell, 8 cells. At the stage of about 150 cells, which means 4 or 5 days after fertilization in men, there is a small ball that is formed with a cavity inside. It is called the blastocyst. And in the blastocyst, there is an agglomeration of cells on one side, which is called the inner cell mass. And already in the beginning of the 80s, Marcus Evans showed that you can disaggregate this inner cell mass and put these cells in culture, and these cells can grow indefinitely. And these are what several laboratories use as embryonic stem cells. These cells are the basis to the production of all the genetically modified mice, for example. And these cells are capable of becoming, once injected into another blastocyst, any part of the newly formed individual. This was shown very elegantly by the capacity of, for example, of an ESL from a black mouse to become, to form a chimera within a blastocyst of a white mouse. It took 17 years until 1998 before the Thompson laboratory in the States showed that you can do exactly the same with humans. So basically, you can do in vitro fertilization, take the inner cell mass, disaggregate the inner cell mass, put this in culture, and these cells, under appropriate stimulation, can become any cell type in the body. Our body contains more than 240 different cell types, and these cells virtually can become any cell type. They are really stem cells. So you'll say, can you take a stem cell, inject it in the heart, it will become cardiomyocytes? And the answer is no. This cell will become a teratoma. So these cells, when put in culture, will become a tumor that is can be more or less aggressive, which is formed by different tissues, where differentiation is very aberrant. These tumors are really very weird to be seen. You cut them, you can see pieces of teeth along with hair and along with muscular tissue, adipose tissue, all together and all in a disorganized manner. However, what you can do is to take the inner cell mass, the embryonic stem cell, and then put them in culture, expand them in culture, and force this cell to become any cell types. This is an experiment that we did a couple of years ago in the laboratory. We take embryonic stem cells from the mouse, we differentiated them into cardiomyocytes, and these cells, you see, they are really become cardiomyocytes. The best proof that they are cardiomyocytes is the synchronous way of contraction. Synchronously in the heart means that there is connexin 43 formed. So one cell is a pacemaker and the tissue contracts well. You can take these cells, take an infarcted mouse, you inject these cells, and you really regenerate the myocardium. Works very, very well. The problem is that these come from an embryo. An embryo has a lot of problems in terms of ethicity of doing this. You know, who is Catholic believes that to get these cells from an individual, you must use in vitro fertilization. You have to reach the blastocyst. Instead of implanting the blastocyst, you destroy the blastocyst, and this is something that for somebody is destroying life. Somebody else who is not Catholic or have different beliefs believes that this is okay. However, there are some big ethical problems, and there is also a medical problem because this embryo comes from fertilization of an egg of a donor with the spermatozoa, could be of the owner, but still the egg must be of another person. So this is genetically, immunologically distinct. So the cells that are generated have a different MHC and different HLA, and so they are recognized as foreign. So this is like a kidney transplantation or a heart transplantation, so the patient must be immunosuppressed. So the question is, can we obtain embryonic stem cells without using an egg for fertilization? And an answer to this came from an experiment that was performed at the end of the 90s by Jan Wilmut, a veterinarian in Scotland, in Edinburgh, who first showed that you can obtain individuals completely identical one another, simply starting from the genetic information of an adult individual. And this was the famous experiment that generated Dolly the sheep. This was not novel, because this is the same concept that was proven by Sir John Gurdon, fantastic embryologist in Xenopus, who already in the 70s showed that you can take a cell from the skin from a Xenopus and then put this cell inside an egg, and this egg reverted the genetic information, started back from scratch, and started to behave as it was a zygote, so a product of fertilization. For several years, people have tried to repeat this experiment in mammals and completely failed, until the Wilmut experiment came at the end of the 90s, and he did something of this kind. He took a cell from the mammary gland of an adult sheep, and then took the nucleus of this cell, took an egg from another sheep, and they put the nucleus inside by an electric pulse, and then this cell, this nucleus here, started to think that he was not a mammary gland differentiated cells, but reverted the genetic information. Now we know that this reversal means removal of the methylation marker of the genome, so resetting the epigenetic information, and then this started to proliferate. At the blastocyst stage, he inserted this cell, this embryo, this blastocyst, in the third sheep, and Dolly was born. Dolly was born 1 out of 243 attempts, which means that the procedure is very, very ineffective. However, this is the same procedure that is now rendered better and used, for example, to clone cattle, to clone pets, and to clone horses. Last year, the International Federation for Sport Races accepted officially that cloned horses could participate in the official competition, just to give you an idea how widespread is this technology in the veterinary field. Why do you want it for humans? To do something of this kind, you take a patient who has, for example, heart failure. You take a cell from the skin, a fibroblast, recover the nucleus, take an egg from a donor, inject the nucleus inside the egg, and then this develops into an embryo. You obviously don't implant the embryo, but simply you get embryonic stem cells. You can grow these cells in culture, and you transform these cells in cardiomyocyte and re-inject the cells back. This has a tremendous advantage towards in vitro fertilization with another kind of donor, because the information here is the genetic information of the patient. It's like if when 80 years before he was a blastocyst, somebody would have taken one cell from this blastocyst, put this blastocyst in the fridge. And then when this cell is needed, taken out of the fridge and cultured again. So you basically recover completely of the problem of need of immunosuppression. This is called therapeutic cloning. You can't do this in Italy because of a law that unfortunately our retrograde country passed a few years ago. However, you can do this in most civilized places in the world. There is still an ethical problem, because to do this, you have to generate a blastocyst, and you have to destroy a blastocyst. And this blastocyst, if implanted, would give rise to a new individual. So for the Catholic Church, for example, this is again something that is not liked. The situation changed very rapidly in 2006, when a group of Japanese scientists at the Kyoto University, fantastic story, the group is headed by Shinya Yamanaka, who is an orthopedic surgeon by training, and found that you don't need to go through therapeutic cloning, so through passage into the egg, but you can just take a fibroblast and inject into the fibroblast vectors that express only 4 genes. These are the 4 genes, 3, 4, SOX2, CMYC, and KLF4. And these genes drive the fibroblasts to become an embryonic stem cell. So basically, you reset the genetic information of the epigenetic information of the fibroblast, and this cell behaves like an embryonic stem cells without having passed through the embryo. So basically here, everybody is absolutely happy. You know that these cells are called iPS cells, and a year later, the Yenish laboratory at the MIT showed that you can use this iPS cell exactly as you would use an ES cell. So you inject into the blastocyst and you get a complete mouse. This is something that excites everybody for any regenerative application. Just a couple of months, a few months ago, well, no, actually, this was last year, went out in the autumn last year, people did the same from skin cells, transform into iPS cells, and then heart cells. And the idea is to move this to the clinics. Producing an efficient amount of heart cells will be difficult. The efficiency of the technology is 1/10 to the -4, 1/10 to the -5 cells, which are transfected with the 4 Yamanaka factors. The only and first clinical trial that is now being approved is in Japan for regeneration of the retina, because the retina is a tissue composed of a little number of cells, small number of cells, so there might be more opportunities for regeneration. The question is, does the heart itself might contain some cells that might promote regeneration? And here, again, there are laboratories that believe that the heart might contain cells for regeneration of the heart itself. However, if you look at the reports in the literature, there are at least 6 different laboratories that claim that the heart itself contains stem cells, but also claim that they have different markers. So it's very difficult to reconcile this. Is it possible that an organ that have one of the least regenerative capacity together with the brain also contains 6 different types of stem cells? There is no answer to this question, but one of the groups have taken these cells, cells from the heart with specific markers, which is the c-KIT surface antigen, and have injected 1 million of these cells in patients last year. The results were reported 2 years ago. In November 2001, the patient seems to do a bit better in terms of function. This is again the Anversa group. I saw Piero Anversa a couple of months ago. He showed me some results showing that they are going to expand this trial. He was very, very confident that this might be working in patients. However, this is very cumbersome because you have to recover cells, purify cells according to this marker, and then inject these cells. And you have to tackle a condition which affects and brings to death 15 million people in the world every year, of which 80% in developing countries. Difficult to conceive that this kind of treatment might be broadly applied to all these patients. The heart certainly contains cells which have a sort of stem cell capacity, because, for example, you can do an experiment of this kind. This is a procedure that Lucio Barile did here in my laboratory, but was invented by an Italian scientist in Rome. And the procedure is very simple. You take a biopsy from the heart of a patient, and then you grow the cells in conditions by which they don't adhere to the plastic, but they are forced to form spheres. And these are called cardiospheres that expand. And this cardiosphere, you see at a certain point, starts contracting rhythmically, which is, again, an indication they have differentiated into cells. This procedure was, as I was saying, invented by Elisa Messina in Rome. Then she collaborated with Eduardo Marban. Then the patent was bought by this laboratory, first in Baltimore, now in Los Angeles. And Marban 2 years ago brought this to the clinics. And there's been a trial by injecting these cardiospheres in the heart. Similar to the sick trial mentioned before, there is some evidence of benefits in the patients. Obviously, the patients are still alive, and there is no way of marking these cells. So nobody, again, knows if the effect of cardiosphere or c-KIT cells, the small beneficial effect, is really due to regeneration or is due to paracrine effect. The skeptics in the field believe that, again, like in the case of the bone marrow 10 years ago, it is that the fact that these cells secrete something that has a small paracrine effect that then fades out over time, that there is no real regeneration. There are, however, some organisms that do indeed regenerate the heart. And for example, if you take the salamander or the zebrafish, you can damage the heart, but also you can damage the limbs, the tail, you can damage the retina, the lens, in the jaws, and these organs are completely regenerated. One of the organs that is really exciting is Axolotl. This is a Maya name because this is a sort of small monster that lives on the mountains, on the volcano mountains in Mexico at 4,000 meters. This is a sort of larval residuate of evolution. You can take this animal and you cut a leg, completely a leg. This is taken from the internet, it's not our stuff. You cut a leg, and then this leg regenerates completely in 40 days. But you can cut 1 time, 2 times, 100 times, and this leg will completely regenerate. And it regenerates the bones, regenerates the muscle, regenerates the nerves, the architecture, regenerates the morphology, the functionality. So it seems that these animals has kept alive also during the adult life the same program that we mammals have had only during our embryogenesis, and then this program is completely off for the rest of the life. So the issue here is, can we somehow reprogram and go back to this original program? The same occurs, for example, in zebrafish. If you cut the apex of the heart in zebrafish, then the heart in 40 days completely regenerates. 2 or 3 years ago, a laboratory in La Jolla asked the question, how does the heart regenerate in zebrafish? Are there stem cells, embryonic stem cells, they come there, or endogenous cardiac stem cells, they go there, regenerate the heart? And they did a very simple labeling experiment by which basically they labeled the cardiomyocyte in the heart, they damaged the heart in zebrafish, and then they asked the question whether the regenerated portion of the heart was formed by these already labeled cardiomyocytes or was non-labeled, so showing that it came from other cell types. And the result was really clear-cut. When you remove 20% of the ventricle, you see complete cardiac regeneration, and all the cardiac tissue that is regenerated is labeled as were labeled the differentiated cardiomyocyte before. So the result is very clear-cut. Heart regeneration in zebrafish comes from already existing cardiomyocytes. If you put this information together with the information of the Nature paper that I mentioned before, that the small cardiac renewal that occurs also in mice occurs through proliferation of already existing cardiomyocytes, it means that there is some residual possibility for already established cardiac cells to go back into the cell cycle and to regenerate. So the question could be here, how can we boost this kind of possibility? And this is the background when we started to work on this about 1.5 years ago. And basically what we thought that, well, we have to reboost the program. So which could be a way to reboost the program? We could try to express some growth factor. We could try to find some drugs. But one intriguing possibility was to use some global regulators of gene function. And the best known global regulators of gene function are microRNAs, because each microRNA, you know, can target hundreds of thousands of genes at a time. So they are global activator of or repressors of entire programs. You know that our genome, in addition to 20,000 genes, and the mouse genome as well, contains about 2,000 genes that code for small RNAs that get processed inside the cell by a variety of biochemical steps until they produce 21, 22 nucleotide long double-stranded RNAs that physically pair with their target mRNAs and depress the expression of the relative proteins, either by inhibiting translation or by degrading this. And pairing only occurs a seed sequence of 7 nucleotides, so they are very promiscuous. They can target a lot of cells. And so basically we took advantage, Anna Eulalio, a very, very brilliant scientist, she's now in Würzburg in Germany, took advantage of the fact that we have this screening facility, the ICGEB, and at that time the known microRNAs, 1.5 years, were about 1,000. We had the library, all the human microRNAs, and so we did a screening for microRNAs that could promote cardiomyocyte proliferation. We debated a lot whether to do the screening with mouse or rat, library or a human library. At the end, we decided for the human library because we wanted something that eventually had a chance to work also in humans. The screening was a screening based on high content microscopy. So you have a microscopic image of cardiomyocyte. These are cardiomyocytes taken immediately after birth from rat. So we wanted conditions where the screening was favorable in our favor, because in rat cardiomyocytes, the neonatal cardiomyocyte contain a proportion of cells that still replicate, as I told you before. So we stain cardiomyocyte in green. We stain replicating cells by EDU, which is the same conceptually similar to bromodesoxyuridine incorporation, and you see this red magenta nuclear cardiomyocyte that replicate. Then the image and these red nucleoside cardiomyocytes are nuclear fibroblasts that contaminate the preparation, so you have an internal control for non-cardiomyocyte induction or proliferation. Then the image is taken up by a confocal, an automated microscope, transformed by the software in a software-generated image, and then we can precisely count in an automated manner how many cardiomyocytes are positive for EDU incorporation. In normal condition, about a bit more than 10% are replicating at day 1 after birth. So we did the screening. We recovered about 200 cardiomyocytes that increased proliferation more than 2-fold. The effect of some of these microRNAs is really impressive. So look at, for example, this one. You see that here in the computer-generated image, the replicating cardiomyocytes have a yellow nucleus, and you see that most of the cardiomyocytes here, where 50% of the cardiomyocytes are positive for incorporation. And then the 200 microRNAs were rescreened in mouse neonatal cardiomyocyte. Remember that the microRNAs of human origin, so we reasoned that if you have a human microRNA that works in mouse and rat, it has a good probability to work also in humans, and then we ended up with 40 human microRNAs that significantly increase proliferation of these cardiomyocytes. Obviously, one question was, is it real proliferation? It's just DNA synthesis, because we measure incorporation of bromodesoxyuridine, so S phase, but if there is mitotic catastrophe at the end, then obviously this will be completely ineffectual. And so we started staining with markers that stain more toward the G2 or the M phase. But I think that instead of showing this, just for the sake of time, I show you this result, which is the most compelling evidence that really we are triggering proliferation. So if you wait 6 days and you give this microRNA, the plate is has plenty of cardiomyocytes that have proliferated and are very happily there compared to the untreated control. We did microarray studies to see what is the physiology of these microRNAs normally, and to our surprise and delight also, we found that a vast proportion of the microRNA that we discovered are microRNAs that are known to be active in the neonatal life, and then they are suppressed in the adult, which is exactly what we want. So we wanted something that brought back a cardiomyocyte to a neonatal conditions when proliferation was still active, and then for some reason is switched off corresponding to the period of birth. Obviously, we wanted microRNA that work in vivo, and so we were very, very keen on testing the effect of this microRNA inside an animal. We started cautiously, so first we took a neonatal animal when there is still some proliferation capacity. We injected the 2 best microRNAs, 199A and 590, directly in the heart with some lipids. And this is staining from bromodesoxyuridine. You see that there is a huge increase in number of proliferating cells. The spectacular things came when we look at these hearts in sections, because these hearts started to be really bigger. You see that this is an injected heart, 2 injected hearts. This is a control heart. You see that the hearts are bigger. What is really impressive is the amount of the left ventricular muscle portion here compared to here. If you look at more closely, well, you can't see much here, but perhaps if you're there, I can see from here. But here you see there are a lot of red dots here. So this the muscle has plenty of cells that are proliferating. Perhaps you can see it a bit better here. This is a magnification, black and white. You see these dotted cells are proliferating cells in a control or in a heart treated with this microRNA. And if you look microscopically, these proliferating cells are really cardiomyocytes that are very happy, seem very happy. They are intercalated with the normal tissue, but they have incorporated bromodesoxyuridine. So really you have production of proliferating cardiomyocytes. For us, it was very easy then to clone the genes coding for these microRNAs inside AAV vectors. So we produced AAV vectors, and this time we put the vectors in the heart, waited a bit more, 12 days. This time the hearts were really very, very much bigger, much bigger. I always say, so I apologize, as somebody has already heard me saying this, that I remember when Anna first did this experiment and came to my office with 2 tubes, and each tube contained a heart, and she told me to guess which is the one injected with microRNA 590 and which is the control. And these hearts were really bigger by eye. And these hearts are bigger not because they are hypertrophic, but because they are hyperplastic. So the size of each cardiomyocyte is identical to the control, but the number of cardiomyocytes is very much increased. And even after 12 days, you see images like this one, that this is an antibody against phosphohistone H3 that marks cells in G2M. You see that marks nuclei of cells that are undergoing division. Is there a therapeutic value for these microRNAs? And so we did myocardial infarction in mice. Serena is a fantastic scientist working on all the animal cardiovascular part. So she infarcted the animals and they injected the AAVs expressing the microRNAs, then observed the first 10 days also EDU was injected to mark the proliferating cells, and then she observed the animals up to 2 months. And this is a parameter, ejection fraction. We measure the percentage of blood which is pumped out of the heart at each normal heartbeat. In humans, about from 50 to 75%. This is a normal level of our mice in this experiment. In the infarcted mice treated with the control, you see a progressive deterioration of cardiac function. This is heart failure, basically. This is a picture of heart failure, exactly as it happens in humans. If you inject the microRNAs, cardiac function is very, very much preserved. At the end of the experiment, we look at the infarct size. Look at, for example, this is the control at 2 months. This portion of the heart here should be exactly like this one, so made of skeletal muscle. Instead, it's made of this collagen tissue, which stains blue with the mason trichrome staining, and this is a big scar. So think of this heart of this animal who has survived for 2 months with a complete scar, replacing the left ventricle. A human would never live with this, such a lesion. Instead, if you look at the lesions in the animals treated with the microRNAs, these infarcts are much, much smaller, and you can find inside these hearts the trace of replicating cardiomyocyte with a nucleus positive from bromodesoxyuridine. We don't know what happens inside these hearts. We try to address this in a omics way. So basically, we took cardiomyocytes, we treated with 2 of the microRNAs, and then we did deep sequences of the transcriptome. We found that there were 640 genes that were downregulated. So this should be the targets of these microRNAs. What's interesting is that among these targets, there is a strong enrichment for contractile proteins. For example, tightening the stiffed proteins in cardiomyocytes. So probably you have to get rid of this protein in order for the cells to divide. And in fact, then based on this result, we took 600 out of these 640 genes and did, and again in a robotic format, knock down one by one of these genes to see if some of these could promote proliferation. And indeed, we found that more than 30 genes individually can promote a bit proliferation. For example, if you knock down titin, where is it? This one, you increase proliferation for, let's see, 5% to 15%. Never to 40%, which is the level of the microRNA. So probably the microRNA works so well because it has several targets, the sum of which increase proliferation. What was most exciting is that these microRNAs not only worked in neonatal cells, but also when we gave them to cells from 7-day-old rats, then these cells were pushed to proliferate. And then we became even more ambitious, and we gave them to adult cardiomyocytes, so the brick-shaped cells that I showed you before. We took these cells, we put them in, these are the brick-shaped cells, we put them in a growth medium. The brick-shaped cells are maintained usually in a medium without serum, so very little serum. So we added 10% serum, they become more rounded, nothing happens. But if you give the microRNAs, these cells start proliferating. So really, these microRNAs seems to work also in adult cells, which is exactly what we want. The thing was really had a good marketing because of, I believe, of the medical need. So Nature wrote a news and views accompanying our article. There was also a coverage by the Nature editors. Last week, the New England Journal of Medicine published a review on our story, and then there were reviews on circulation research, on molecular therapy, Nature Biotechnology. This week here in Italy, Le Scienze, who is the major scientific journal for the lay public, dedicated the cover to this discovery, which fills us of responsibility now, obviously. We have to do something except doing basic science and understanding how this really works. So the question for us is, where do we go from here? So how we can translate this discovery in something useful. And we have started working on several steps to transform some of these microRNAs into something that possibly could go to humans. So we have to formulate the microRNAs. We don't want to use AV vectors for this application. I wouldn't like to have an AV vector expressing a microRNA pro-proliferative for the rest of my life. And so basically we are going to use them as naked RNAs, but we have to chemically modify them to render them more stable and to formulate them in order to facilitate penetration inside the cells. And then in collaboration with Fabio Recchia in Philadelphia, in Pisa, we will test them in larger animals for efficacy. And obviously we want microRNAs that trigger proliferation, but of the cells that we want they trigger proliferation, so cardiomyocyte. We don't want unspecific proliferation, for example, of cancer cell or other cell types. And there's a long way to go, but we are very concentrating in somehow trying to see if this might become a real therapy. The way I see this possible therapy is a person has a myocardial infarction, and then this person is brought to the interventional cardiology room. If it is brought there within a few hours from the infarction, then usually he gets angioplasty. And this opens up his arteries. However, if a portion of the myocardium is dead, this will become a scar. So after a few days from that, 5 days, 7 days, the same interventional cardiologist reaches the coronary artery and inject a bolus of the microRNAs that should get into the cardiomyocytes still living, bordering the infarcted region, and promote the regeneration. Even if this is very partial and unsuccessful, there is just a proportion of this proliferate, still you recover a portion of contractile tissue, which in clinical terms can really make a real difference in the long term. A lot of people to thank again. I've mentioned their names, I think, all through the talks, so I don't need probably to repeat them. I do this rapidly since we are recording that. So Serena does all the animal experimentation and coordinates all the animal work at ICGEB. Miguel runs the manager of the high-throughput screening facility. Anna did the screening for the microRNAs on the molecular studies. Lorena produces the AAV vectors. And Matteo Del Ferro is a clinical cardiologist who is also an excellent experimenter. So he spends half of the time in the clinics, half of the time in the laboratory. Fantastic example of a real MD, PhD type of work. And we collaborate with the clinical group of Gianfranco Sinagra for this work. And again, I thank you very much for your attention.